Assalamu alaikum, guys, and good evening. Before I start, I just want to tell you guys that today I've just tried another method of sending you guys all the invites, those who have already joined the Google Classroom link. So I send the invites to your email, and I can see that most of you guys were able to join the class without being waiting for me to allow you to enter the class. So I think that's going to work well. So in this way, there will be less disturbance during the class if someone comes late. I hope so. Okay, so what we studied yesterday was the process of DNA replication, and we have gone through in detail about the process of replication in prokaryotes and the eukaryotes. What we're going to study today is the remaining part of this central dogma of molecular biology, which I mentioned to you guys during the intro lecture of molecular biology. And these are transcription and translation. So what we're going to do in this presentation, if we would be able to finish this, inshallah, in today's class, so we'll be covering these two topics of transcription and translation. Now, going back to the point, as I said, that the central dogma of molecular biology is that flow diagram, which is just in front of you, where you can say that there's a process of replication, which is mainly for the DNA or the genetic material of an organism. The need of this DNA replication is obvious because every organism is growing, cell multiplication is going on, whether by binary fission in prokaryotes or by meiosis in the eukaryotes. So the cell multiplication is going on, and as a result of that, you need to multiply the genetic material as well. So in this case, DNA replication or the replication of the genetic material is a must process associated with every cell. Now, from the DNA, there is another domain where we go towards the final product of what is being coded on the genes. And that final product is called proteins. Yesterday, we discussed about a lot on mutation. And during the mutation, we have been discussing that any change on the DNA sequence, which might result in the change of the genetic code, which in turn results in the change of proteins, is going to have some impact. Sometimes they are going to transform into genetic disorders, and sometimes there might not be any phenotypic expression of that mutation. So the ultimate goal of the genetic material, which is encoded on the DNA, is to get proteins out of that, which is the final product. So the sequence of that is, we start from the DNA. The first step is transcription. The transcription is the formation of RNA molecule, specifically messenger RNA molecule, from the DNA molecule. And then from that messenger RNA molecule, the next step is the translation, which involves the other two type of the RNA as well, which were told to you earlier on, transfer RNA and ribosomal RNA. And as a result of the translation, we got the final ultimate product, that's the protein. So this is all about this flow which is in front of you is being referred to as central dogma of molecular biology. Yesterday we have discussed about a couple of enzymes and couple of proteins and their role and function in the process of replication. So today we're going to discuss some more enzymes and proteins and discussing how the transcription and translation takes place. Before I go in the detail of transcription, there's a process I need to discuss a little bit, and that's being called as a reverse transcription. I think the name itself is self-explanatory that this process is a reverse process of transcription. Now, what actually mean by that reverse process of transcription? As I said, the transcription is the formation of RNA from the DNA, and in most cases, it's messenger RNA. However, there are few exceptions to the central dogma, which I just showed you in the previous slide. Most importantly, some RNA viruses, their genetic material is RNA. They don't have DNA. So some RNA viruses called retroviruses, this is one group of RNA viruses, and they make a DNA copy of themselves using that RNA, which is their genetic material. So in this case, a DNA molecule is being copied or made by using an RNA template. This process is called as reverse transcription, and the enzyme which is involved in this process is called reverse transcriptase. The DNA copy incorporates into one of the chromosome and becomes a permanent feature of the genome. The DNA copy is inserted into the genome, that is called a provirus, and this represents a flow of information in the direction from RNA to DNA, unlike the normal flow which is from DNA to RNA. Now, closely related to some of the retroviruses are another group which is retrotransposons. These retrotransposons are the sequences of the DNA that make RNA copies of themselves, which then get reverse transcribed into DNA. So this is also a process of reverse transcription, but in a little bit of different way. These DNA that insert into new location in the genome, and unlike the retroviruses, retrotransposons always remain within the cell, and they lack the genes to make the proteins that coat and surrounds the virus. So they are not going to transform into a provirus, but in case of the retrovirus, they are transformed into provirus. That's the difference. Another important term that I want to discuss with you before we go into detail is prions. I'm not sure that whether you have heard of this term before or not. A prion is an infectious protein. Now, when we talk about the prokaryotes, we know that prokaryotes can be of few cells, or they might be unicellular. Smaller to a prokaryote or a unicellular organism is a virus itself, which is not a complete cell, we know. And then smaller to the virus are being called as prions. These are only protein, but they are capable of causing an infection. So in short, prions are called as infectious proteins. These prions are the agents that cause, for example, mad cow disease, which is bovine spongiform encephalopathy, causing chronic wasting disease in deer and in many other animal. I'm not going into detail at this stage. These diseases, what they cause actually, they cause the neural degeneration in humans, the damage to the neurons. And the symptoms are approximately those of Alzheimer's disease. You might have heard of that. That's the degeneration of neurons that takes place in Alzheimer's syndrome. And that's why this particular neural degeneration caused by the prions are same in terms of the symptoms as Alzheimer's. So that process is accelerated to go from onset to the process till death in about a year. Fortunately, the disease is very hard to catch and very rare, and they usually have a long incubation time. There is no cure known for this yet, and not enough is known about how it's spread and how to prevent this. The prion proteins, which we call the PRP, they're normally present in the body like all proteins. They are folded into a specific conformation, a state called PRPC, which is prion diseases. And these prion diseases are caused by the same protein folded in an abnormal way, unlike the normal protein. So now you got the understanding that prions are the infectious protein. How they became infectious? These proteins are in the body, but somehow because of their wrong folding or misfolding or problem in their folding structure, they become infectious and causing infections. So these are called prions. RNA, we all know, we've studied about the RNA, that RNA is a single-stranded polynucleotide chain, a central role in the life of the cell. And we see mostly its role in protein synthesis, but there are many other functions of RNA as well. RNA can be a genetic material information storage molecule, just like you studied in RNA viruses. But yeah, most of the cases, the genetic information is being carried out in DNA. One theory for the origin of the life has it starting out as RNA only. This is one concept, that how the life originates was actually from RNA. Then adding the DNA, that means the reverse transcription, as I mentioned, and then proteins later. This theory is being referred to as RNA world. Now, the RNA can make a hybrid structure with the protein. And that hybrid structure, they are involved in protein synthesis, which are being called as ribosomes. I already have discussed about that earlier on in the intro lecture. The splicing of the messenger RNA, the telomere maintenance, guiding ribosomes to the endoplasmic reticulum, and many other tasks. Recently, it has been found that very small RNA molecules are also involved in some sort of gene regulation. Gene regulation is just like you studied in 411, the metabolic regulation. So just like that, the whole process of this replication, transcription, and translation also requires regulation, and that's called gene regulation, which will be our next topic after this lecture. Now, the RNA, as I said, primarily is used for the protein synthesis. And we all know about the three types of the RNA, so I'll very quickly go through that. Messenger RNA, which is a copy of a particular gene or more than one gene, which is located on the DNA molecule, and that particular gene corresponds to a particular protein. So when we need that protein, we need to make a copy of a messenger RNA from that gene on the DNA. And then it will go on further to be translated, and you'll get the protein out of that. So groups of 3 bases in mRNA are called codons. You already have got idea about that. And each of the code is having specifically for amino acids, and we have 64 codons. 3 of them are the nonsense or the stop codon. 61 of them are the functional codon. And there are 20 amino acids, so you have more than 1 codon for 1 amino acid. That's what we have studied already. In eukaryotes, the initial RNA copy of the gene which is being made is called primary transcript, which is then modified to form mRNA. What does that mean? It means that when there is RNA synthesis from the gene on the DNA, the first copy of the RNA is being referred to as a primary transcript. And that primary transcript will undergo some modifications, some changes, and after that, it will become a mature mRNA molecule. So that means there are stages from the gene to the mRNA. The other 2 types of the RNA are the ribosomal RNA and transfer RNA. We already studied that ribosomal RNA is the structural RNA involved in the formation of the ribosomes. It's a hybrid of RNA with the protein. There are 4 different RNA molecules that make up the part of the structure of the ribosome, and they actually perform catalysis of adding an amino acid to the growing peptide chain. Or you can say they are the machinery, they are the site where protein synthesis takes place. The third type is the transfer RNA. It's being referred to as translator RNA as well. This is a small RNA molecule which acts as an adapter, because that's the molecule that will read the codon of the messenger RNA and will bring up the respective amino acid for it. So these are the 3 types of the RNA used in protein synthesis. When it comes to the difference of RNA and DNA, there are few differences that you are already aware of, and we have already gone through that. RNA has a sugar ribose, DNA has deoxyribose, RNA contains uracil, DNA contains thymine, RNA is single stranded, DNA is double stranded, RNA is short, smaller in size, one gene long at most, and DNA is long, containing many genes. Okay, let's start the transcription. As I already said, the transcription is the process of making an RNA copy of a single gene. Now, why we are saying again and again that is the process of making RNA? Because transcription involves the synthesis of all the three types of the RNA, mRNA, tRNA, and rRNA. All of them are synthesized from the DNA as a result of the transcription. But in generally, when we talk about transcription, the RNA molecule that first come into our knowledge or mind is mRNA, which is right up to some extent. But the tRNA and rRNA are also being transcribed from the DNA molecule. So it's basically a molecule which is as a result of a single gene, specific region of the DNA. The enzymes used in transcription is called RNA polymerase. It's a homologous enzyme, just like the DNA polymerase you studied yesterday. There are several forms of RNA polymerases as well. In eukaryotes, most genes are transcribed by RNA polymerase 2. Just like for the DNA replication, for RNA synthesis, you also need the raw material. Like you need free nucleotides, and there are four ribonucleotides which are required for RNA synthesis, which are ATP, CTP, GTP, and UTP. It's instead of TTP, which is for the DNA. It's the same ATP as it's used for the energy in the cell. As with the DNA replication, the transcription also proceeds in the same direction always, which is 5' to 3'. So a new base is added to the 3' hydroxyl group of the growing chain. Unlike replication, transcription does not need to build a primer. Now, that's a very important point to be kept in mind. You remember last time for the DNA replication, there was a need of a primer because DNA polymerase was not being able to catalyze the reaction right from the beginning. But that's not the case with the RNA polymerase, so you don't need a primer. Instead, transcription starts at a region of the DNA which is called a promoter. So that means the gene which we need to transcribe must have some sequences which we call the promoter. These promoter sequences will be identified by RNA polymerase enzyme, and thereby it facilitates the binding of the enzyme on the right location to transcribe that gene of interest. For protein coding genes, the promoter is located a little bit away, a few bases away to the upstream. The first base that is transcribed into the RNA. That means if we have a gene, for example, which uh, corresponds to a particular protein, then the promoter region will be a little bit away from that particular gene because gene needs to be transcribed properly and then it needs to be translated properly to the protein. But the promoter itself is not the part of the gene, it's a little bit away from, a few bases away from the gene sequence. The promoter sequences are very similar to each other but not identical. So if many promoters are compared, a consensus can be seen or can be derived. All promoters would be similar to this consensus sequence, but they have little differences. And these little differences are very important. Like if we say that all the promoter sequences are 100% identical, then it doesn't make sense because then how are we going to differentiate which gene needs to be transcribed? So that means there is a pretty much long consensus or similarity in the sequences of the promoters, but with certain uh, necessary necessary unique sequences which differentiate one promoter from the other promoter. Now, in the process of transcription, the transcription starts with the RNA polymerase enzyme, as I said, binding to the promoter region. The result of this binding, it only occurs under some conditions. And what are these conditions? When the gene is on. What does that mean? The gene is on means when your body specifically require a protein, then there are certain signals being sent to the DNA molecule to activate that particular gene for which we need to synthesize the protein. For example, if there's a particular enzyme which is needed by the body, we know that proteins are enzymes, so their coding will be on the genes. So the requirement of that enzyme actually will trigger and will turn on the gene. That's what I mentioned earlier on, called the gene regulation. So there, the gene should be turned on. That means the message has been conveyed that we need to transcribe that gene. Various other proteins and other transcription factors will also help the RNA polymerase bind to the promoter. Other DNA sequences further upstream from the promoters are also have some role. Now, once it is bound to the promoter, the RNA polymerase will do the unwinding, which was being done by the helicases in case of the DNA replication. So the binding of the RNA polymerase to the promoter will trigger unwind, but this unwind is not just like in DNA replication, a continued process. This unwind will be for a small section of the DNA. Which section of the DNA? That particular section of the DNA upon which the gene is located. We don't want to unwind the entire DNA molecule. We just want to unwind that particular part of the DNA where our gene of interest is there. And once it is unwind to that region, then this DNA unwind region will act as a template, and we need to synthesize an exact RNA copy of that particular part of the DNA. The DNA strand used as a template is called the coding strand, and the other strand of the DNA will be called as the non-coding strand. Now notice that RNA is made from 5' to 3', just like the DNA replication. So the coding strand is actually read from 3' to 5. So the template strand is read from 3' to 5', and the newly synthesized RNA will be parallel, that will be 5' to 3'. Now the RNA polymerase will proceed just like the DNA polymerase proceeds from starting point to the point of termination. So RNA polymerase proceed down the DNA and will keep synthesizing the RNA copy. In prokaryotes, each RNA ends at a specific terminator sequences. The terminator sequences are those specific sequences which will be identified by the RNA polymerase as a point where we need to stop the process of process of transcription. And thereby, at this point, when it reaches the terminator sequences, RNA polymerase will be separated out from the DNA. In eukaryotes, the transcription doesn't have a definite endpoint. The RNA is given a definitive termination point during RNA processing. So there's a difference in the prokaryotes. The termination takes place with the specific sequences, while in the eukaryotes, the RNA is synthesized a little bit longer than what actually we need. And then that's why I just mentioned earlier on that we need some sort of processing after the RNA is being made to make a final mRNA out of it. Now this is transcription graphics, and you can see that we divided that into 3 stages as initiation, elongation, and termination. So initiation is the binding of certain proteins which facilitate the binding of the RNA polymerase at a particular promoter region, results in the unwinding of that particular region of the DNA, and this is the template, and this is the newly synthesized RNA. As it goes down, it will synthesize that particular gene, which is our gene of interest. And once it reaches specific sites where it gives the message that the process is complete, this RNA polymerase will be dissociated. You got an RNA copy, and these hydrogen bonds will again join together to reunite it, and the DNA would be in its original intact form. Now, after transcription, as I mentioned in prokaryotes, the RNA copy of the gene is messenger RNA itself. There is no such process is needed further to make it mRNA, because it's just being copied with the what actually we want to, and then this mRNA is ready for the translation. So in fact, in the prokaryotes, translation starts even before transcription is finished, and I'll explain it later on. In eukaryotes, the primary RNA transcript, the first copy of the RNA which is being made, which might have certain sequences of the gene which is transcribed, which is not required. And that's why it needs further processing. And that particular process of processing after transcription is called post-transcriptional modification. So this post-transcriptional modification is a particular characteristic of eukaryotic transcription process. And then afterwards, we all know that in eukaryotes, this process of transcription is going on within the nucleus, and nucleus has a definite membrane. So you need to have a transport mechanism for this, finally processed mRNA from the nucleus going outside to the cytoplasm, where it will be utilized for the protein synthesis on the ribosomes. While in case of the prokaryotes, we don't have a definite nuclear boundary, so that's not the step required in this case. So there are 3 steps of the RNA processing, which I call post-transcriptional modification. And what these 3 steps are, don't forget that this is for the eukaryotes. The first step is adding a cap to the 5' end. The molecule of the RNA which is being synthesized recently need a cap structure on its 5' end. What is cap? I'll go through in detail. Then there is a poly A tail, that means adenosine, adenosine, adenosine at the 3' end. And then the third one, which is very, very important, is splice out introns. So let me go in the details of these 3, specifically the third one, to get you an idea that what this post-transcriptional modification is. The first thing is the capping. Now look at the structure. This is the structure of the cap. The RNA is inherently unstable, and there are many reasons for that. And one of the reasons is that it is a small single-stranded molecule. So that's pretty much unstable, and its half-life is very small. So we don't want this mRNA or RNA, which is recently transcribed, to be unstable or degraded before it's translated to protein. So during this process of RNA synthesis till the protein synthesis, we need to stabilize that RNA molecule. And in order to stabilize that, we need a capping. The ends are modified, and still it's not only the capping but also the tail. They both play the role in the stability of the RNA. So at the 5' end, there is a cap structure which is a modified guanine. It's not an ordinary guanine, it's a modified guanine called 7-methyl guanine. So on the 5' end, we have specifically 7-methyl guanine, which is not an ordinary guanine. It's a modified guanine, and that is called as cap. And then at the 3' end, we have a poly A tail. And at the 3' end, the primary transcript RNA is cut at a specific site, and a poly A tail of about 100 to 200 adenine nucleotides are attached. 100 to 200 adenine nucleotides at the 3' end. That's called the poly A tail. Note that these A's are not coded in the DNA of the gene. So that's pretty much clear that this cap and this poly A tail is not specifically the part of the gene that we want to translate. These 2 are the modifications specifically being given to the RNA to make it stable molecule, and there are other functions which you will come to know later on. The third modification in the post-transcriptional modification was the removal of introns. Now let me tell you the 2 terms. The one term is called intron and the other term is called exons. Now the word exons comes from exit, while the introns, the term comes from inside. What happens that in eukaryotes, when the RNA is being transcribed from the DNA, remember yesterday we studied that on the DNA of the eukaryotes there are regions where the genes are located, and then there are the regions between the 2 genes called intergenic region. Now on the intergenic region, we do have nucleotide sequences, but they do not correspond to any gene. So when the RNA is being transcribed in eukaryotes, or in other words, when an RNA copy is being made from the DNA in eukaryotes, it is transcribed in a way that some of the genes are transcribed, as well as some of the region which is not the part of the gene is also transcribed. And remember that part which is being transcribed as an extra part, which doesn't correspond to any of the protein, it must be removed. It's not the part of the final product. So before this process of translation takes place, we need to cut down those extra part of the transcription machinery, which are not the part of our final product of the protein. Those extra part are called as introns. Why we call them introns? Because they will be cut and then they will be left inside the nucleus before the mRNA is being transported out. While the other part, which is I said exons from exterior, is that particular sequence which is transcribed for that particular gene, and this is what is being transported as a mature mRNA from the nucleus to the ribosome for final processing as protein synthesis. So introns are to be cut, left inside, and exons will be transported out. So exons actually correspond to the particular gene region, while introns are the non-genetic material region which needs to be cut down. Now, there are few prokaryotic examples of this intron and exon, but it's very rare. Primarily, this concept of intron and exon is found in eukaryotes. Some genes may have long introns. So like the dystrophin gene, which the mutants cause muscular dystrophy. It has more than 70 introns that make up more than 99% of the gene sequence. Only 1% is that gene is the one which is coded for protein. The 99% of the gene sequence is comprised of intron. So it might be the case like this. However, not all eukaryotic genes have introns. So, just make it a balance that sometimes there are introns part of the gene and sometimes they are not a part of the gene and sometimes they are a bigger part of the gene. So in short, we need to remove that intron before this mRNA is going to be processed for protein synthesis. So this is called the intron splicing or cutting. Introns are removed from the primary RNA transcript while it is still in the nucleus. The introns are spliced out by RNA protein hybrids called spliceosomes. This is again a kind of enzyme. The intron sequences are removed and the remaining ends are reattached to make a final RNA consist of exons only. So you can see here primary transcript, and this is how it cut down the portions, and then this is the mature RNA molecule. So that's how the introns are removed and exons will be rejoined to make a mature mRNA. This is another example of how it goes. This is the gene on the DNA, and you can see here that it's mentioned here this intron A, intron B, exon 1, exon 2, exon 3. So as a result of the transcription, this is pre-mRNA and it's just copied like a DNA. It has intron and exon both. Then the capping takes place as 5' end. Then the splicing takes place, introns are removed, only exons, and finally the poly A tail at the 3' end, and that gives you the mature mRNA, which is ready to be transported to cytosol on the ribosome for translation. Now, we come to the translation now, and that is how the final product we will get, and that's protein. Now, proteins are composed of, you have already studied one or more polypeptide chains and additional small molecules called cofactors. The polypeptides are linear chains, but they undergo different 3D modifications and changes and, hydrogen bonding and folding, and that gives rise to a functionally mature 3D tertiary or quaternary structure of the protein. The sequence of amino acid is actually what determined by the gene sequence, which is actually the primary structure of the protein. So amino acid and peptide bond, I'm not going in much detail because that's not the topic of interest at this stage, because you already have gone through that, but just quickly going through that. There are 20 amino acids, and every amino acid must have an amino group and a carboxylate group, and then there's the R group, which is the variable group, and two amino acids are joined together with a peptide bond, and there's a removal of water, and then so on. polypeptide chain grows and water molecules remove, and that results in a polypeptide chain, which is what I said, the primary structure of a protein. These are the 20 amino acids which are being shown here. Now come to the process of translation. The translation of this mRNA, now remember I said that transcription involves formation of not only mRNA, but also rRNA and tRNA, but we are not going to discuss about that. Now, talking about the translation of mRNA into a protein is accomplished by ribosome, which is itself an RNA protein hybrid. And ribosomes are composed of two subunits, a large unit and a small unit. And that's different in eukaryote and prokaryote, as I told you in the intro lecture of molecular biology. The ribosomes bind to the translation initiation sequence on the mRNA and then move down the RNA in the direction of 5' to 3'. So when the mRNA is attached with the ribosome, and afterward the reading frame starts and starts reading the sequences of the mRNA in the 5' to 3' direction, thereby getting amino acids which are according to the codons, and then these amino acids will be joining together through a peptide linkage to make a polypeptide chain. So every attached amino acid will have a free amino group for the next coming amino acid, and the last amino acid in the polypeptide has a free carboxylate group which will be left free once the protein is synthesized. So each group of 3 nucleotide in mRNA, we know that it's called codon, which codes for one specific amino acid. Transfer RNA has the role, I told you already, they have anti-codon. They actually read the codons on the mRNA and they will bring about specific amino acid. So we have specific tRNAs for specific amino acids, and that's how they bring up new and new amino acid, and these coming amino acids get joined together to form a polypeptide chain. This is the structure of a transfer RNA. It's a cover leaf-like structure. It has different loops. This loop is called amino acid loop, where the particular amino acid is attached. They have anticodon. That's the one that's attached with the mRNA to read the code. And then there are other, sections of this tRNA, which are not, pretty much important at this stage. But you can see that it looks like a cover leaf structure, but make sure to see that this is a single stranded molecule. It's not a double stranded, although there are hydrogen bondings at certain areas, but this molecule starts from this point, goes like this, and it will end up with this. So it's not a double stranded structure. It's a single stranded structure, but it has certain complementary hydrogen bondings, which resulted in these loops to be formed. So the transfer RNA molecules are short RNA that folds into a characteristic cloverleaf structure. Some of the nucleotides are modified to become things like pseudouridine and ribothiamidine. Each tRNA has 3 bases that make up the anticodon. These bases pair with the 3 of the codons of the mRNA, as I said, to read the reading frame. Each tRNA has its corresponding amino acid I just mentioned to you, which is attached on its 3' end. A set of enzyme which is called amino acyl tRNA synthetase is actually the enzyme which are used to charge the tRNA with a proper amino acid. Some tRNAs can pair with more than 1 codon as well. The third base of the anticodon is called the Wobble position. These 3 anticodon, one of that is called the Wobble. That means it can be utilized for more than 1 base pairing. So that's why if this, for example, if it's TAA, it codes for a particular specific amino acid. If it is TAC, it can code for another amino acid. So this one change of the anticodon is called the Wobble position, which allows one tRNA molecule to base pair with more than 1 mRNA genetic code. Now, for the initiation of the translation, in prokaryotes, the ribosomes bind to specific translation initiation sites. Let me think about I think I should stop here at the initiation, because otherwise it will be a little bit complicated at this stage. And inshallah, we are going to complete this, translation process in our next class, hopefully. So I'm going to share the presentation with you shortly as well. So now I'm going to open the forum for questions. So if you have any question, please raise your hand and ask the question. Any question, guys? Okay, so if there is no question, I'm going to finish the class and stop recording, and I will share the recording with you guys once it is processed. Inshallah, thank you very much. See you in the next class. Khuda Hafiz.